All right, folks. I think we have waited enough. Others can join in as they wish to. Good afternoon, everyone, and thank you for joining us for this melon industry RD&E update. Obviously, we have within our various projects components and requirements for us to share and bring updates to the broader industry as we go along. And we figured we'd take the opportunity today to grab roughly an hour of your time to give an update across 4 different projects. I'm Jonathan Davey, the CEO of Melons Australia. Today we'll kick off with a bit of a presentation by Cherie Gambley on the identification and management of a fungal disease complex in melons, project VM 22001. Then we'll hand across to Dr. SP Singh to give us a run through on the melon food safety monitoring and support phase 2, VM 24001. And then Chanjeev Chandrakumar on strengthening sustainability of melon production, VM 23001. And we'll close it out with Mark Lufin, who's going to present on the melon quality improvement program, VM 23002. Just for everyone's information, we are recording the session today solely because that will then give us something that we can utilize to put up online for those who can't be here, to continue sharing the wealth of knowledge that we're all going to experience over the next 45 minutes or so. We'll look to wrap it up at about 2:45 Eastern time, and then we'll have about 15 minutes of question and answer. So if I can please ask, if you've got questions throughout, just jot them down on a piece of paper, and when we hit the end, we'll get through the timing and the presentations, and then we'll open it up for Q&A, or stick it in the chat and we can deal with it at the close. So thank you, Cherie. I'm going to hand over to you so that we can kick off with ID and management of fungal disease complex in melons, please.
Thanks, Jonathan. Hang on, get my mouse working. Okay, can you see this?
Yep, certainly can. Just need to hit the presentation button. All good. Done.
All good? Yeah.
Yep.
Cool. Okay, so our project covers a few different diseases, mostly fungal ones. Today I just want to talk a bit about gummy stem blight and anthracnose and what we consider to be the major risk factors for disease outbreaks. So first of all, gummy stem blight. This is a fungal pathogen we used to call Didymella, but now that species been split into 4 different species, 3 of which infect cucurbits. And the three that affect melons are Staganosporoscus, Cucurbitacearum, and Citrulli. Those 2 species have definitely been linked with gummy stem blight disease outbreaks. The third one is known to infect melons, but it's never really been found in the field as a pathogen-causing disease. In the US, there's a lot of the Citrulli species. And in Australia, we don't really know which one we've got. We know we've got all 3, but we don't know which is the important one in the field here. But by the end of the project, we should have a better idea of that. And the photo on the right shows you the fruiting bodies of the gummy stem blight on a melon stem. So I'm sure many of you are familiar with the symptoms of gummy stem. You're getting lots of lesions. They can be tan, brown. You get them on the leaves, the petioles, the stems, the crown. Sometimes you get a brown gummy exudate coming out of cracking, particularly on the crown and the stems. And you get, as the disease develops, you get black fruiting bodies present on the infected tissue, and they can be quite diagnostic, although they can look a little bit like other diseases like anthracnose and charcoal rot. And you can get vine death in really severe infections, and that's typically linked with where the crown or the stem cracks really badly. The vine can't fill fruit, but it has to be pretty severe to get that happening with late infections. So this is anthracnose. It's an Colotoptricum fungal species. There's supposed to be multiple races in watermelon. Again, we're not sure what we have here in Australia. The symptoms are similar, and the fruiting bodies superficially look like gummy stem with the black spots on the stem. But when you look under a hand lens or a microscope, you can see these little fingers sticking out, and that's very diagnostic of anthracnose. This is to me, this disease is most severe when it's affecting the little seedlings, and you get this damping off, and you can get really high mortality due to anthracnose in really young seedlings. So talking about gummy stem blight, what causes infection and disease spread? So the key with gummy stem blight is it's all about vine wetness. So you need an hour of vine wetness to get infections, and then the longer the vine stays wet, the worse the disease becomes. The optimum temperature is between 20 to 25, but you can get disease outbreaks outside that. So temperature is not critical. It's more the vine wetness that's the key factor. And obviously high relative humidity has a role, because that means basically the vines take longer to dry out. And the pathogen is also spread in water and wind. And the picture on the right shows a chain of conidia, which are the fungal spores, and they get released like that in water, in a drop of water. And so you can get millions of these conidia coming out in a few drops of water. And so there's a lot of the pathogen there available to spread. And this is a particular risk to new plantings if they're close to older crops. So the two really high-risk sources of the pathogen for the disease development are old crop debris and infected seedlings. So anthracnose, it's important to note here, it develops and spreads in under similar weather patterns, and it's common to have both of them together. So we wanted to do a bit of an infection risk profile to make it easier to understand what the real risks are. And we've broken this down into the source of the pathogen and the weather. So again, the cucurbit crop residues and infected seedlings are the two main sources of the pathogen that you need to be aware of. We'll come back to this picture in a minute. I just wanted to talk about this old crop debris. So we did some field trials here in Bundaberg where we planted into old plastic and we planted into new plastic. And you can see the photo at the top is the old plastic where there's a lot of vine still on top. We had 100% seedling mortality in that block 3 weeks after we planted, whereas below you can see that there's still a really nice plant establishment in the new plastic, and we had no mortality in that block at all. The disease incidence in the new plastic got close to 100% towards harvest, but the plants were still producing fruit quite happily, whereas the old plastic, we had nothing left after about a month. We also did a second trial where we looked at delaying planting into that old plastic to see what would happen over time. And as I said before, immediate replanting back gives you 100% seedling mortality. If you wait 2 months, the mortality isn't high at all, like we didn't lose any, but the disease incidence was 50%. And if you wait another month and don't replant until 3 months later, the disease incidence goes right down to 9%. So it's really important to manage that old crop debris. And in our instance, I think one of the key factors in removing that old debris was just wind in the environment in that 3 months. I would also say here that we're talking about anthracnose and gummy stem in this field trial. A lot of the seedlings in that old plastic were actually died due to anthracnose. So the other important factor is the weather, and in particularly rainfall events or anything that keeps the vine wet. So in 2023, we did some monitoring over time of a rockmelon crop. We did a few crops. This is an example of a rockmelon crop on the left. And you can see at the first monitoring date, we had very low incidence of disease. Most plants were healthy. And then there was a significant rainfall that happened in early May. And you can see the disease severity has then shifted quite quickly. Within 2 weeks, from mostly being healthy to mostly being mildly affected. And then we also did a watermelon crop at the same time. And again, the first monitoring date was sort of mid-March. And most of the plants had a mild disease incidence or disease severity. And then we had a rainfall event in mid-March. And again, within 2 weeks, that disease severity had shifted to moderate and severe. So rain and vine wetness has a significant effect on disease development, and it's all about timing. So if you get the wet weather early on, you're going to have more problems. If you have wet weather later, you may have no problems at all. So this brings us back to our risk profile again. And so we have the cucurbit crop residues, and you can see here, if you have old crop residues and extended periods of vine wetness, your risk profile is right up in the high zone. Similarly with infected seedlings, if you have that extended period of vine wetness. And then as you have less wetness, your risk lowers. And overall, your risk is much higher with cucurbit crop residues being still in place when you're planting compared to infected seedlings. Infected seedlings are a significant source of disease, particularly if you get a lot of them. But typically that's less pathogen load in the field than what you get with old crop residues if they're still around. So how do we manage these diseases? Well, we talk about three key points, I guess. Use quality seedlings. We're all well aware of that. Reduce that crop debris prior to planting. That'll be a significant one. And then obviously you need to use fungicides strategically, and that all depends on what other pathogens you have in the field as well. Integrated disease management is always really important, and there is a strong overlap with anthracnose and gummy stem blight, just with weather patterns and pathogen sources. So you always need to manage those ones together. Just as an extra piece of information, we did do a variety trial here at Bundaberg on watermelons, but unfortunately we couldn't find any genetic resistance or tolerance to either anthracnose or gummy stem blight. Thanks.
Thanks, Cherie. Amazing timekeeping. And of course, that final slide there about the Horn Innovation Melon Fund, all of these projects are funded by the Melon Fund. So thanks. I know Sharon and others are online for everything you guys do in supporting this. Now throwing over to Dr. SP Singh about the Melon Food Safety Monitoring and Support Phase 2. Over to you, SP.
Thanks, Jonathan. Can you see my slides?
Yep.
Yeah, all good. Thank you.
Great. Good afternoon, everyone. I'm going to talk about melon food safety. As we all know, it's really critical to have fresh produce, which is safe for consumption. If there's contamination on produce, it can make people sick, or even if the contamination is found on produce in the supply chain, product recalls also cost a lot to the industry. So our project is part of the Safe Melons program. So I think what we are really trying to achieve in this project and program is to stay out of the news. In 2018 was the last Listeria outbreak in the melon industry, and that also led to the start of Safe Melons program at national level, and it's 7 years since we had any recalls or any incidents related to melon industry. So credit goes to growers who are adopting best practices to manage their food safety risks. So our program is highly focused on melon growers. It's national in scope, and on an average, we engage 150 melon growers in the country, and the program is now internationally recognized for its achievements. So in this project, the phase 2 of melon food safety monitoring and support, as you can see, there are two components, monitoring and support. In monitoring, we are monitoring food safety practices of growers and doing targeted surveillance for various foodborne pathogens like Salmonella, Listeria, and sugar toxin E. coli. And then we are capturing the data on food safety, using it, sharing within industry for continuous improvement. As part of our support component, it's really important to support growers in risk mitigation practices. And if there are any issues that arise from time to time, we have to do rapid and targeted response. And we also do a lot of outreach activities and technical support to growers. So as part of our surveillance and monitoring, we collect microbiological samples throughout the supply chain. We collect fruit samples from growers, packers, wholesalers, and retailers. And then a lot of environmental samples are also collected from packing sheds, cool rooms, equipment from processing and field equipment, and then soil, water, and whole range of samples we collect and analyze for various pathogens. As I mentioned, in 2024, we engaged with almost 150 growers across the country. The map shows the location of melon growers, and it's really critical to know where these melon growers are and what do they do and how do they grow melons and what are their food safety practices. So since this project started in January 25, so in last 3 months, I would say from January to March, we captured 55 growers from 5 states and Northern Territory. This is a busy slide, but just to let you know that in 3 months, we collected approximately 345 samples from primary production, processing, wholesale, and retail. We try to capture all types of melons and from different regions of melon production. And during these 3 months, we had a pretty rough start of the year and the project. You can see some of the extreme weather events that happened in the last 3 months. And I'm going to talk a little bit more about them. So when we collect samples and we analyze and we occasionally come across some pathogens, one of the case study I would like to share is that in February 2025, we had multiple detections of Listeria from environmental samples from a packing facility, but there was no detection of Listeria on fruit. So in response, grower initiated the deep clean and sanitization protocol, and this is how the response is undertaken. But fortunately, no melon fruit processed during that week and afterwards showed any positive detections. So where did we find Listeria? It was on the bin tip platform in the processing shed, and then on the conveyor belt in the wash water runoff, and few detections from the conveyor belt. So we investigated and tried to do root cause analysis. This is what is done in the project as part of follow-up investigation. We found that we did sampling on 12th of February, but there was a rainfall in the previous week, 19 mils. It doesn't sound much, but February is a pretty dry month for that particular region. So during sampling, there was visible mud on the fruit surface that potentially brought Listeria from soil into the packing facility. So despite having that high organic load on fruit, Packer was really effective in managing risk. They had very high levels of sanitizers and wash water, appropriate washing, brushing, and contact time. So as a result, there was no detection of Listeria on fruit surface. So we closed this investigation, and then the grower and the packer, they have been doing pretty well since then. And in March 25, as most of you recall, that there was a leafy salad recall. So I got a lot of inquiries from growers regarding sugar toxin E. coli, what it is. And we published an article on sugar toxin producing E. coli. So just to briefly explain, it's basically a subgroup of E. coli. So E. coli, which contains STX sugar toxin genes along with some binding factors, they are really serious pathogens which can cause human illness. So STEC in our Safe Melons program, I think melon industry was the first in the country to have a look at STEC. So we have been looking at sugar toxin E. coli since 2021. Initially, we were only looking for strain 157, which is most common, causing human illness. But now we look for all top 7 STEC serotypes in our regular surveillance. Normally, STEC comes from healthy cattle, sheep, and wildlife, and contaminated water and animal feces are the major carriers. We generally do not have STEC outbreaks in Australia, unlike other countries, especially US. So though the STEC notifications are increasing, but all those notifications are pretty sporadic in nature. So the question is, do we get STEC in melons? Yes, melons are grown up in the field, and we see detections on melons, very random, and they're sporadic in nature. Two factors which are favorable to melon industry: drip irrigation and plastic mulching. But normally when we get STEC detection on melon, it's heavy rainfall prior to harvest, it's proximity to cattle and grazing before melon production, or wildlife incursion. Interestingly, after the cyclone, ex-cyclone, I should say, in Southeast Queensland, we increased our surveillance activity, and from 11th of March onwards, there was heavy rainfall in Southeast Queensland, and we collected samples from markets right from 17th of March onwards. And we had 3 detections of sugar toxin E. coli on watermelons. And there was just one random in each sampling event. But it was really interesting to note that the fruit was harvested on 14th of March, but it could survive until 24th of March on the fruit. So we went back to the grower and we looked at their production systems. I think it was a shortage of watermelons, and they went back to their farm and they picked some pollinizers. As a result, there was some contamination coming from wildlife incursion in a part of the property. However, this grower has been very active in terms of wildlife management. This is their infrastructure investment from last year. They put a fence to exclude pigs from their property. And you can see on the right some of the pig footprints outside the fence. So the system is working, but it needs to be made better. So that is one part of the story. And the second is food safety help desk. We engage with growers and other supply chain partners since this new standard has kicked in now. There's a lot of engagement with regulators. So I had meetings with regulators in Tasmania, South Australia, New South Wales, and Jonathan presented Safe Melons work to Queensland food safety regulators. However, we receive a lot of technical inquiries from growers across the country, and most of the technical inquiries are around primary production and processing standards, extreme weather, and also STEC in last 3 months. We also provide support to new growers and packers. So late 24, 2 packers recommenced rock melon operations, one after 5 years, another after a short break. Another grower or a packer has established packing facility in Western Australia late 2024, and 2 more packing facilities are in the pipeline. So from our project, we provide them technical support on how to set up systems which are food safety friendly. Once the systems are set up, we do verification of their washing, sanitization, and the whole processing sort of operation that they undertake. So as I said, dust storms have been around and impacted a few melon production areas. You can see on the slide, this is the normal dust path in Australia. So starting from Carnarvon through to Sunraysia and Riverina, that's the dust path, which is common, and windbreaks growers use to minimize the impact on melons. But those windbreaks, sometimes they are not really effective. So a grower shared some of the pictures from a dust storm. You can see even if there were windbreaks, they will not be very effective in reducing the dust load. So these dust storms are really powerful carriers of pathogens and whole range of contaminants. So I would like to conclude that food safety monitoring and management, they're critical for the industry's sustainability and profitability. And extreme or even moderate weather events such as rainfall, bushfires, dust storm, they impact melon food safety. And the project team responds to these extreme weather events to make sure that we assess the risk and help growers and manage those risks. So the top 5 practices that we always list for growers and packers that are really very unique to the industry. So no use of untreated or raw manures in melon production. Melons, especially which are washed, they should be pre-cooled before washing. Drinking quality water for washing and preferably run to waste washing systems. and washing and sanitization with appropriate sanitizers and contact time, and finally cleaning and sanitization of harvest equipment and packing facilities. So these are top 5 practices that we always reiterate and tell the industry to not forget about these. So going forward, this phase 2 of the project, we are again aiming for 0 product recall and 0 food safety incidents, and we will continue to engage with the industry to make sure those food safety issues are dealt on time and in a very targeted manner. And I would like to thank Melons Australia for organizing this webinar and Hot Innovation for funding. And big thank you to all the melon growers and packers and even wholesalers who are engaged in this project, because without grower engagement, such programs would not be successful. Thank you.
Thank you, SP. Very insightful stuff. Now, if you can stop sharing your screen, we'll throw over to Sanjeev to give us a run through on the strengthening the sustainability of melon production project. Over to you, Sanjeev.
Thanks, John. Thank you for the opportunity. So as John mentioned, I'll be giving a brief of what we have been doing with Melon Australia and the growers lately. So in this project, we are trying to tackle the challenge of developing a sustainability story. Like we know there are a lot of environmental issues, as SP was showing before, and farmers, including melon farmers, are having tough time to face these environmental crises. So that's one side. And on the other side, we also see that as an opportunity for growers and the industry bodies to act in advance so that we have the useful tools and techniques in order to evaluate the environmental impacts and take necessary measures to address any hotspots that we think very critical. So in that context, our project tried to develop an environmental performance benchmark for the melons industry, focusing on key environmental impact indicators like carbon emissions, water usage, and eutrophication. And this is a 2-year project. At the moment, we are running a pilot study with selected pilot grower groups to identify key data points and assess the key environmental impact performance for their farms. So what we have done to date is to identify the pilot and then run the pilot study to develop a benchmark. And the expected outcomes of this project is an Excel-based calculator, which will be hosted by Melons Australia or Hot Innovation, so that farmers in future, they can provide the data into that calculator and generate the results for their environmental performance. It will also show what are the major hotspots, so those insights can be used for them to develop relevant solutions to address those environmental impacts. And as I mentioned before, at the end of the project, we try to develop that sustainability story for the melon industry to say how good they are and what they're doing at their best to make sure the Australian melons have the right story to tell both locally and internationally. As a deliverable, we are intending to develop this kind of a one-page outcome for each of those growers who are participating in this study. So this will have all the information for each grower, like where the farm is located, what size was the growing season, and what are the key environmental impact performance for their farm. And they will also highlight what are the hotspots so they can start working on addressing those hotspots. So the pilot study to date, we have engaged with 5 growers across 3 states already, and we have done particular farm visits and collected the key data points in order to develop the calculator. So we are currently developing an Excel-based tool or calculator using one of the database called EcoInvent, which have environmental impact results for different life cycle processes like manufacturing, transportation, different chemical use, etc., etc. And once we complete the pilot study and the tool is developed, we will generate the results for each of those pilot growers and provide the results and show what are their hotspots so they can start using the tool as well as the results to improve their sustainability story or improve the environmental performance of their production system. As going forward, we want to do the same analysis at the industry level so that we can develop an industry-wide benchmark. But it's a challenge because we need more growers to come on board and provide data. So one thing I want to re-emphasize that all this data which are being given by the growers are always anonymized, and then which means we make sure the confidentiality of the data is always preserved. And if required, we are happy to sign NDAs during the data collection process. So as the final outcome, we'll then develop a sustainability story, not only for these pilot groups, but also for the entire industry of melon growers. And also we are thinking of delivering a short training program so that they know how to use this online tool once it's hosted by Melons Australia or Hot Innovation, and also intend to do a wider stakeholder engagement webinars to disseminate the outcomes of this project. So moving forward, what we need actually is, like, if there are any growers on the call today, on the webinar today, we would like to have a chat with you to get some more data about your farming practices so that we can use your data to validate our calculator and enlarge the sample size which we have at the moment, so that can also benefit for us in order to make sure our model or the calculator is robust, and also make sure the results which we develop for the industry is more representative rather than just focusing on a smaller sample size. So if you are interested, please reach out to John or Joanna or myself. Our contact details are here. Drop us an email. Thank you for your opportunity. I'm looking forward to the questions. Thanks, guys.
Thanks, Chanjeev. Much appreciated. And well done, everyone, for keeping us on time. Now, Mark, I'm going to throw to you as the lucky last presenter before we get into some questions to brief us on the Melon Quality Improvement Program, please.
Excellent. Thank you, Jonathan. What's up? Cool. Melon quality improvements. So where are we? We'll start with an executive summary. So from the data coming out of Nielsen IQ, melon sales are up. On-farm sampling and measurement protocol is now available, and that we've got some tools in behind to do harvest prediction, and that's already showing benefits for growers. And our work on non-destructive maturity assessment is currently being trialled alongside the wholesale and retail inspections. What are we here for? Basically to improve consumer demand and value for seedless watermelon, rockmelon, and honeydew. And so how are we doing? For the Nielsen home scan results for the year ending 23rd of February this year, you see melons are up in terms of dollar sales and in terms of volume. The most interesting thing to me is, if we're looking at the dollar sales, is actually look at where the major supermarkets are down compared to the 8.9% overall, which we see from here. And it's actually the other supermarkets and the non-supermarkets that are really pushing forward there. Watermelon sales are up, and again we see a similar pattern with the other supermarkets and non-supermarkets leading the charge there. Rockmelon dollar sales are up just very slightly, and again, we see the major supermarkets are actually pulling us down, whereas the other supermarkets and the non-supermarkets are doing pretty well. In terms of compliance, we're seeing over the last 3 years, seasonally, watermelons are pretty static, which is a little bit of a concern. Rockmelons are heading nicely. If we look season 22, 23, 63 to 70 to 81, where our target is 80%. So from that perspective, we see with the major retailers, independents and wholesalers, all looking good on rock melon. Honeydew has been the poor cousin for a while, starting off in the first season at 24%, next season 34, 49, heading for 70. So we're certainly heading in the right direction with the honeydew. Inspection reports, we have inspected over 1,000 melons now, and whenever we can identify a grower, if you've given us your contact details, we'll send that report directly to you. That's probably within 24 hours of that fruit being inspected. If you haven't given us the details, we'll send it through to Jonathan, who will forward that to you. So why are we doing this? Selling fruit below standard damages the industry, and research shows that after a negative eating experience, consumers don't repurchase for at least 6 weeks. Some are out for the rest of the season. So if we look at 60% in the brown, we see, and compare that to 80%, if 100 consumers come a week later, 80, a week later, 64, you see there's a 50% change. And with the work we did with Citrus Australia, one of the supermarkets had strong sales, repeat purchase and category growth directly linked to what we did with them. On farm, sampling and measurement, we now have a sampling and measurement protocol available on the Melons Australia website under the Melon Quality Control Project. So have a look there. That is available to all growers, free download. We have been working with some cell phone apps, and especially if you have staff doing this, they can indicate where they have sampled the fruit for you as a farm manager can see, have they done that sampling properly? We've got graphical outputs that shows anything in the green is good, in the yellow is acceptable, and the red's not good. You can see here, this particular sample, there's a number of fruit on the red that clearly the fruit need a bit more time. We can model this and do harvest predictions. So with the target there in the red, we're looking at the purple model. We model all the fruit in the block, and that's proved pretty successful up till now. We can also graph out your weights, and so you can see this has an average weight of about 2 kgs, but anything from about 1,500 to 8, and also model out the growth in weight. And you can see here that as the water gets dropped off near the end, the weight starts dropping off a little bit. Non-destructive maturity assessment is well underway now. Over a period of months, we've tested a whole bunch of models on various watermelons, rock melons, and honeydew. Now we've selected 3 models, cantaloupe, honeydew, and mango models, and we're using those in Melbourne and Sydney alongside the standard inspections. So that data has just been collected at the moment to see how that pans out. So an overall summary, melon sales are up, especially in outlets other than the major supermarkets. So that's a challenge for us to speak to the major supermarkets. The on-farm sampling and measurement protocol is now available on the Melons Australia website. That's already providing benefits to growers that we've been working with. Harvest prediction is now available, and as with Change Chief, we'd be very happy to work with you. Come and have a chat. Inspection reports are now being, as soon as we can find a grower in the monitoring, we can send you an inspection report. Non-destructive maturity assessment is being trialled at the moment. We're doing a lot of work at the moment and just bringing that to a conclusion on exactly where to measure a Brix in a melon, and that's coming to a conclusion. And we've also been able to measure transport shocks on farm through processing. And whenever we're on farm, we often get requested for that. So does this stuff work? Here's some work that we have done with citrus in New Zealand. And you're seeing before we started, very similar compliance to what we're seeing in watermelons at the moment. And this has been the last virtually 10 years. And you can see the last 4 years, nothing less than 95%. So do we need to move the standard? No, the industry picks that up really well. So thank you very much, everyone. Back to you, Jonathan.
Thanks, Mark. Very much appreciated. So now we have roughly 18 minutes left on the clock for any questions. I'm gonna kick it off because I've got the mic at the moment. For others, if you want to raise your hand just up the top of the screen, and we'll come to you in order of how quickly you can get it up there. I had one for Chanjeef in particular. So you mentioned in the presentation that you've had 5 growers engaged to date. Where and what do you think the sweet spot is in terms of number of growers for being able to create some conclusions on sustainability that we can then start to advocate and put out there?
Yeah, thanks for the question, Joe. So at the moment, we don't have a representative of the country because like most of the growers come from Queensland, like 3 from Queensland, 1 from Western Australia, and 1 from Victoria. So if we want to develop this sample size to represent Australia, we need more samples, like more growers coming from other states as well, so that we can develop like a country representative sample. And also in terms of the production volume these growers produce, they only represent a smaller volume of the national melon production. So if we can aim to find a few large growers in the country, then we can cover a quite considerable proportion of melon production in the country.
Brilliant, thank you. For SP, there is a question in the chat from Susanna, which is, you showed a map of dust storm pathways. Is there increase in monitoring of these areas as part of testing and or additional guidance for validation of washing? And what do you do in terms of response support post-event?
Yes, that's a great question. We keep a track of these weather events in respective melon production regions, and we do increase testing of product from wholesale markets. But by now, growers and packers, they understand the risk, and they are also increasing their like washing capacity or sanitization methods, so increasing the sanitizer concentration and increasing the contact time, or having a bit of a liberal amount of water on the product during washing and sanitization. So yeah, so there are a whole range of things that growers do to manage that risk, and we also increase frequency of testing and the number of samples during those events.
Excellent, thank you. Any questions from the floor? I can't see any hands up, so Cherie, I've got another one for you. In terms of some of the work you're doing, we're just wondering, is there going to be any trials run in terms of management options, and how do we start to, I guess, address some of these issues as they move forward? I know you had your couple of dot points there around how to deal with it at the moment, but what does it look like long term, and what management options might be considered?
Thanks, Jonathan. Yeah, so we're starting doing more field trials now in the project, and we're starting with things like cover crops and other ways to reduce that pathogen load in the fields. So I was talking today about gummy stem and anthracnose, but charcoal rot has really emerged as a significant issue for melons. And there's also fusarium in the mix as well. So we're trying to look for a way of, reducing that population of all those pathogens down in the field. And that's going to be a range of field trials, starting with cover cropping, and other ways to use maybe some biological control agents that help the old debris to be decomposed faster, so that risk of replanting is lowered. We're also doing a lot of work with in vitro fungicide testing. So I mentioned charcoal rot. There's nothing registered for charcoal rot in anything that it affects, and it affects quite a few crops. So we're doing quite a bit of in vitro testing of fungicide actives to see if we can find something that will work on charcoal rot. And similarly with the gummy stem, we're also doing a lot of in vitro trials to look at different fungicides. There's some interesting ones that came out of the US which affect the life cycle of the fungus so that it doesn't produce those fruiting bodies. So you don't get those millions of spores released. And I think something like that could be particularly helpful, and especially for nurseries, to stop the spread within the nursery with the seedlings, to stop that sort of pathway. So there's quite a lot going on. Does that answer your question, Jonathan?
I think it's I think it's a good summary of what's going on, what we can expect, and where the project's going in the future. Thank you.
Unfortunately, we don't have genetics. Like, that's always the gold answer for plant diseases, if there's genetics in the varieties. But so far, what we've tested hasn't really shown any promise against, with tolerance or resistance.
Oh, question for you, Cherie, from Neil from UPL. Is there being developed a fungicide BMP for the GSB complex rotation of? I can't even say that. Can you read the question in the chat?
Hang on, let me see.
Dithiocarbamates, Jonathan.
Go.
Oh, dithiocarbamates, like mancozeb.
Yeah.
Yeah.
So dithios, systemics, SDHIs, strobilurins, etc. Is there a rotation frequency, etc.?
Yeah, so we haven't sort of moved forward with any best practice management guides in relation to fungicide rotations at this point. The project's really trying to get a handle on what may or may work, and then I guess you can go forward with how you might combine that. And, because there's multiple pathogens, but you don't always get all of them, you would need to modify those practices to suit, I guess. I don't know if we will come up with a best management practice guide for all of them at the end of the project, but we'll definitely have recommendations on what I would see the agronomists that advise growers specifically as having more of that role on how to combine products within their region, because there's such site specificity around some of this stuff, and local timings and things like that are pretty important. So I think our role is more to find the chemicals that do and don't work, and then help with the agronomists in how they might deliver that. I don't know. I think with things like this, a best management practice guide is almost out of date by the time that you've printed it, because something will have changed for sure. Did that answer your question, Neil?
100%, Sherry. I just asked Temo the question. I thought I'd go to the source, so you answered it better than Temo.
Oh, I'll tell him that.
Thanks, Neil. Jürgens, have a question on the line from you? Fire away.
Ms. Gambley, just maybe to follow up on what you just answered, besides the chemical practices, and you said there's been some look at the variety resistance and genetics. Does that include rootstocks or not?
So we haven't done rootstocks yet. The watermelon varietal trial we had was just the varieties, and that was for anthracnose and gummy stem. And unfortunately, there wasn't any resistance. Len Tesoriero, who's also on our project, does a lot of work with the fusarium pathogens in the project, and he's done race typing with those pathogens, and he is looking to look at some of the rootstocks for that, because at the moment the rootstocks are pretty important with fusarium if you want to grow in a fusarium affected block, and we're not 100% sure how they're holding up with the races that he's finding. So that'll help for that. With the macrophamina, the charcoal rot, I think it is useful to have a look at the susceptibility of some of those rootstocks to that pathogen, because nobody's looked at it. Nobody has looked at charcoal rot in melons really at all. So anything's open for consideration to try. And I have no idea whether they would be susceptible or not, but I think it would be a good thing to try.
Yes,
that's particularly for watermelons, obviously. I think there is some rootstock and grafting in rock melons, but it's not as much as it is in watermelons.
Yes, no, but maybe adding to that, because if you, for example, had a paddock like you showed with the replanting of already used field, that it could just be maybe a more strategic use to do, so, say, for example, your direct seeded rock melon, and then the next season you plant grafted watermelon, which would not be as susceptible. But as you said, no, it will only be determined with further investigation. I'm just thinking practically, because as you mentioned earlier rightly, with a good management practice guide, diseases also evolve, not just our solutions. So
yeah, that's right. And some growers choose to use grafted plants depending on the season as well to get better establishment under some seasons. So yeah, I think there is scope for the rootstocks.
thank you very much. I might have a next one for Dr. Singh on the food safety. Doctor, I've tried to see on some would there be more value in post-harvest treatment of fruit to keep preventing any disease spread? And if there's maybe a chemical, I don't know how many people are familiar with a product called Spore Kill, chemical active. I know it's internationally used sometimes or not. The dicyl dimethyl ammonium chloride, which could maybe act as a double action for both treating both fruit and your equipment. Could that perhaps mitigate further risk, being a bit more proactive, especially if you had dust or rain events, or if you're using it on your packing line, so it both sanitizes your fruit and your equipment at the same time?
Jürgen, are you talking about watermelons or rock melons?
Any fruit.
I think with rock melons and honeydews, we need to make sure that any sanitizer that we use should be food grade. So the product that you mentioned, I understand, is not food grade. So for post-harvest,
melons, no, not on melons, on other crops, but would it be worthwhile investigating? Because again, I'm just asking out of experience, because back home where I'm from, it was listed on much more crops to be able to be used both as a spray with a tractor spray rig and inside a pack house. My question would be, would it be more worthwhile to see if there's more options like that?
Yeah, no, it would be interesting to look, but my only concern is that anything that is used post-harvest has to be food grade. So that is the requirement. So we only use food grade disinfectants, which is chlorine, peracetic acid, chlorine dioxide, or even ozone in wash water. So yeah, so some of the products, I mean, the product that you mentioned is good for cleaning equipment and also facilities. But we need to differentiate those two, food grade versus non-food grade products. So the industry uses disinfectants, which are APBMA approved. So yes, it's for pre-harvest application, it would be different. I'm not sure whether that is registered for pre-harvest use on the crop in melons, but yeah, I think
no, it's not.
Yeah, so we need to be
Not in
very careful
Australia.
that any chemical that we use, it has to be food grade if it comes in direct contact with melons, especially after harvest.
So just a comment, Ben Coombe from Seven Worlds, we sell spore kill. So it is registered in post-harvest for inedible peel, tropical fruits. So yeah, it can be used. It is registered for that use.
Thank you.
Yeah.
Thanks, Ben. Great insight. We'll take that one offline for a bit further discussion, I reckon. Any final questions? No. Well, I'm going to give everyone 3 minutes back. Thank you all. Thank you, Mark, SP, Cherie, and Chanji for the presentations and for the question and engagement session. Really appreciate everyone coming along today. As I said, we have taken a recording of this. We'll make sure that it is put up online and shared more broadly. So please feel free to go back and review it. And any further questions, we can certainly make the contact details of our presenters available for everyone. Thank you all. Much appreciated. Have a great Tuesday afternoon, and we'll see you again soon. Don't forget to register for the Melon Conference coming up Wednesday, the 4th of June in Brisbane. Pre-Hawk Connections. We'll see you all there.
Thanks, Jonathan.
Thanks,
Thanks,
Jonathan.
everyone.
Cheers.
Cheers, guys. Yeah.
Thank you.
Bye-bye.